She leads the neurophysiology of human movement group and 2 large multidisciplinary teams of researchers that are investigating early diagnosis and motor signs in Parkinson's disease and the long-lasting effects of methamphetamine on movement. So I'd like you to please welcome Associate Professor Gabrielle Todd as she shares with us about her research project involving investigating early diagnosis and motor signs in Parkinson's disease.
Thank you everybody for this lovely opportunity to speak with you today. What I'd like to do first is to give a brief explanation of how the brain controls movement under normal circumstances, and then I'm going to go on to explain what happens in Parkinson's disease and some of the work that we've been doing in this area. Now, many of the movements that we perform every day happen without us really having to think too much about it. And this suggests that movement's a simple brain function, but actually it's not. It's really quite complicated. If you take this particular example here, the ability to grip and lift an object requires a grip force that's enough to overcome the effects of gravity on the object and prevent it from slipping. But that grip force can't be too high, otherwise you'll damage the object that you're trying to hold, and you may cause yourself unnecessary fatigue. The brain also needs to make some quite complicated calculations when you go to lift an object that you've never encountered before. And these difficult calculations are trying to essentially work out how much grip force is going to be required to lift the object. And this calculation is based on the physical properties of the object, our prior experience, and the individual's safety margin for error. The brain also needs to predict in advance what's going to happen during each phase of the movement, and it does all of these types of calculations without you being aware of it. Because of all of these sorts of calculations and processes that need to take place, the control of movement involves a large proportion of the human brain. And when there's damage or disease to any of those movement parts of the brain, it leads to what's called a movement disorder. Now, movement disorders typically are characterised by the types of symptoms that present. Some examples of symptoms are having an abnormally large tremor, or rigidity, which is excessive joint stiffness or muscle tone. Some people might experience tics, which are repetitive, stereotyped movements or vocalisations. Other movement disorders have problems with posture and walking, and other movement disorders have problems with performing movements at an appropriate speed. Now, some movement disorders are only characterised by 1 or 2 of these types of symptoms, but other movement disorders have a whole range of symptoms. And when there's a whole range of symptoms, those movement disorders can be difficult to diagnose because often the symptoms overlap between movement disorders. Parkinson's disease is one movement disorder that is characterised by a lot of symptoms. The symptoms of Parkinson's disease are an abnormally large resting tremor, rigidity. There's also problems with balance, and people often have a shuffling gait. And people unfortunately perform movements at an abnormally slow speed as well. Now, Parkinson's disease is a disease that predominantly affects older adults. And unfortunately, we don't know what the cause of Parkinson's disease is. There's also no definitive diagnostic test at the moment, which leads to a quite high misdiagnosis rate. So up to 1 in 5 people with Parkinson's disease are misdiagnosed. Unfortunately, there's also no cure either. The current treatments just try to reduce the severity of the symptoms and thus try to improve quality of life. Now, there's been quite an enormous effort dedicated to research on Parkinson's disease, and that research has fallen into 5 broad themes. So trying to get a better understanding of what the underlying disease process is, how we can protect neurons from further damage and disease. There's also a research effort dedicated to trying to improve the treatment and also identifying people that are predisposed or at risk of getting Parkinson's disease. And there's also a pretty big research effort dedicated at improving diagnosis and early detection. What I'd like to do is just spend a minute or so to talk about the underlying disease processes that occur in Parkinson's disease before I start to talk about our contribution to this research effort. Now, Parkinson's disease is a disease that affects, initially, the parts of the nervous system that are involved in our sense of smell and the neurons and the nerves that control our intestines. So this leads to the presentation of the earliest symptoms, which is problems with our sense of smell and gut motility, and in particular constipation. Now, these two parts of the nervous system are actually the parts of the nervous system that are exposed to the outside world through the things that are in the air that we breathe in and things that are on the food that we eat. And because these two areas of the nervous system are affected first, it's thought that Parkinson's disease actually involves exposure to some environmental factor in genetically susceptible people. What happens next is that the disease processes move from the parts of the nervous system that are involved in our sense of smell and control of the gut up to parts of the brainstem. And it's when the disease processes move their way up to the brainstem where we start to see the movement symptoms occurring. And the reason that we get the movement symptoms occurring once the brainstem becomes involved is because the brainstem has a very important brain region that's involved in movement, and that brain region is called the substantia nigra. So what happens in Parkinson's disease is there's a significant loss of neurons in that brain region, the substantia nigra, and this is what leads to the movement problems starting to appear. Once these movement problems appear, that's what prompts people to go and see their doctor and then to subsequently receive a diagnosis of Parkinson's disease. But by the time that the movement symptoms have appeared, the person has already had Parkinson's disease for 5 to 10 years before that. What happens next is that the parts of the nervous system that are already affected by Parkinson's disease get progressively worse, and then the disease processes move up to other parts of the brain. And when it moves up into other parts of the brain, we can get some additional symptoms that involve problems with memory and thinking and emotion. So this whole disease process occurs over many, many years, and in some instances, decades. Now, I have two main interests when it comes to Parkinson's disease. And my first interest involves identifying people that are predisposed or at risk of getting Parkinson's disease. And this research involves looking at the appearance of the substantia nigra. So that was that brain region that was really important for moving, and it's the brain region that's primarily responsible for the movement problems in Parkinson's disease. Now, what my colleagues and I did is we actually had a look at the appearance of the substantia nigra in a group of young people that had used the drug methamphetamine or ice. Now, these two topics seem quite different to one another, but they're actually not. They're related. And the reason that they're related is because methamphetamine and Parkinson's disease affect the same set of neurons. They both affect what are called dopaminergic neurons in the brain. So methamphetamine is highly toxic to these neurons, and we happen to have a lot of those neurons in the substantia nigra. Parkinson's disease is associated with loss of those neurons. Those neurons die. Now, methamphetamine is actually so good at damaging these neurons that it used to be used as an animal model for Parkinson's disease. So what I mean by that is scientists used to give methamphetamine to laboratory rats to study the processes that might underlie Parkinson's disease. So given this, it's not surprising that then perhaps you might think, well, maybe young people that use methamphetamine might be causing damage to this particular brain region, the substantia nigra, and putting themselves at much greater risk of actually getting Parkinson's disease. And that's what my colleagues and I showed or demonstrated. In a world-first study, we showed that people that have only used methamphetamine as little as 5 times have changes in the appearance of the substantia nigra, and these changes are the same types of changes that we see in older adults that actually have Parkinson's disease. So here's some data from that particular study. On the y-axis, we have the appearance of the substantia nigra, this brain region here. viewed with transcranial ultrasound. And what that measure is the area of bright signal that's in this particular brain region. In the people that had used methamphetamine, only as little as 5 times, the area of bright signal in the substantia nigra was significantly larger than 3 other groups of participants that were in the study. The other groups of participants were people that had never used illicit drugs before and people that had used 2 other types of illicit drugs like ecstasy and cannabis. But it was only the people that had used methamphetamine that had the abnormality. Now, this abnormality is quite clinically important because this abnormality that we saw in these young people was the same abnormality that we see in older people with Parkinson's. So 90% of older adults that have Parkinson's had the same brain change that we were seeing in young people that had used methamphetamine. So this was quite an alarming result from our study because Australia has a massive problem with use of methamphetamine. 1.3 million Australians have used this drug, which is a big number considering our total population is only 24 million. Because of this, we've taken a lot of our research findings on this risk of Parkinson's in this particular group, and we've translated those research findings into a new public health campaign to try to educate young people about the effects of this drug on movement and movement parts of the brain and risk of Parkinson's disease. And hopefully you'll see some of the advertisements associated with that campaign in the middle of next year. So looking at people that are predisposed or at risk of getting Parkinson's disease is just one of my research interests. My other research interest involves trying to improve diagnosis of Parkinson's disease and early detection. So this is work that we're currently doing at the moment, and it also involves looking at the appearance of the substantia nigra, but this time using MRI. So here's an MRI image of the brain that enables us to view this particular structure that I'm interested in, the substantia nigra. So on this MRI image, the very top of the image is the front part of the brain, the back is at the bottom of the image, and then we're looking at the right and left sides as well. In the centre of the image is a butterfly-shaped structure, and that butterfly-shaped structure is actually the top of the brainstem, and you can see that butterfly-shaped structure in the top image. Within that butterfly-shaped structure is the substantia nigra, that brain region that's primarily affected by Parkinson's. And the substantia nigra looks like 2 stripes on the butterfly's wing, and you can see those 2 dark stripes on the top image. If I magnify part of the MRI image and make it a little bit larger, we can see the butterfly-shaped structure outlined in orange, and the 2 dark stripes there and there that are the substantia nigra. And this is the image taken from a healthy older adult. Parts of the substantia nigra appear dark in colour on an MRI, but there are also some parts that appear lighter in colour as well, or a white colour. Now down here, I'd like to show you the image of a person that actually has Parkinson's disease. So in a person with Parkinson's, we see a change in the appearance of the substantia nigra. One of the big changes that we see is the loss of the light-coloured areas in the image that are taken from people with Parkinson's disease. Now, because of the changes that we can see on the MRI in people with Parkinson's disease, it's been proposed that MRI of this particular brain region might be a good diagnostic aid for Parkinson's disease. However, there's a bit of a problem with this technique, and the problem is that it involves a lot of subjectivity. So at the moment, what has to happen is a radiologist looks at the image and then needs to make a judgment whether the substantia nigra looks normal or not. And this is quite difficult to do because radiologists don't get a chance to see many people that have a normal substantia nigra. And this is because most of the people that get referred for that type of imaging are suspected to have Parkinson's disease. Not only is that a problem, but we don't have any sort of reference data about what the substantia nigra should normally look like in healthy people. And this is quite important because you need some type of reference point to compare against. So in our research, what we're doing at the moment is we are seeking to remove some of the subjectivity from the process by creating an objective method for assessing this brain region, but then also providing some normative data about what this brain region normally looks like in males and females and in people of varying ages. Because then this data can be used to compare against. So radiologists can compare what they see in a patient to what then is known to be normal, and then make a more informed decision as to whether the image is abnormal or not. And if we can get to that point, then we hope that that might aid diagnosis of Parkinson's disease. So our current research study is really trying to look at these two potential solutions to this existing problem. Our current research study involves using MRI to measure the substantia nigra and to measure the characteristics of the substantia nigra in 100 healthy adult volunteers. So these 100 adults are aged from 18 to 80, and what this information will tell us is what the substantia nigra should normally look like across ages and in males and females. And this would provide then the reference data that radiologists need to make the comparisons against patients. Our study also has 35 patients that are currently living with Parkinson's disease. And we've included the patients in our study because we need to make sure that the technique, the new objective method that we've developed, works, and that we can see significant changes between people who have Parkinson's and those that don't. So we've included the Parkinson's group as a comparison. What our current study is also doing is trying to improve understanding about what this brain change means on the MRI images. And so what we're doing is we're actually also looking at what we call functional correlates of this brain change. So what I mean by that is we are actually looking at the appearance of the substantia nigra and then trying to relate it to how people's nervous system are actually functioning. The function that we're particularly interested in is movement. And so we're assessing movement in this study in various ways. One of the tests involves a neurological examination performed by a neurologist that specialises in movement disorders. But we're also doing some laboratory-based tests as well that give us more detailed information about movement. Some of those laboratory-based tests involve picking up key-shaped pins and putting them in corresponding holes. We also attach a little gadget onto people's fingernails to measure tremor. And we also ask participants to grip and lift this little gadget, which enables us to assess how people plan to perform movements and how they actually perform the movements through controlling the various muscles that are required. In our study, we're also looking at speech. And the reason we're looking at speech is the ability to say a given word requires the fine control of over 100 muscles in our body. So speech is a very complicated and specialised form of movement. We're not necessarily interested in this study in what people are talking about. We're actually interested in how they're saying the words, how they're using and controlling their muscles to make speech words or speech sounds. In our study, we're also doing some tests of memory and thinking as well. And the reason that we're doing that is because we'd like to know if we see changes in the substantia nigra, early changes there, are they associated with early changes in other brain regions that are involved in memory and cognition? So it's looking to see whether, if we can pick up changes in the substantia nigra, is that indicative of some other changes that are occurring elsewhere in the brain and the way that the nervous system is functioning. So through this particular study, what we're really hoping to do is to overcome some existing problems for using MRI to help diagnose Parkinson's disease. And the way that we're trying to overcome those problems is to make it easier for radiologists to use and to remove that, some of that subjectivity that's currently involved in the process. We're also hoping to give radiologists the tools that they need to make more informed clinical decisions And part of those providing the tools is to give radiologists the reference values that they need to compare against. If we can solve some of these problems that exist with using MRI in the diagnosis of Parkinson's, then hopefully we can get to a more accurate diagnosis of Parkinson's and to be able to diagnose Parkinson's earlier than is currently possible. So I'd like to finish by thanking all of the people that are involved in the research that I do in Parkinson's. I haven't listed them all here. On this slide, I'd just like to particularly thank the people that are involved in our current project on the diagnosis of Parkinson's disease. I'd also like to thank the organisations and individuals that have kindly supported our research along the way. And I'm very happy to answer any questions that you may have about Parkinson's disease or our research. And we are still looking for volunteers if anybody is interested in participating in this study. Thank you. Thank you. Thank you.
So has anybody got any questions for Gabrielle? First up is Grant.
Hello.
Thanks, Holmes. One question I had was with trying to get objective measures of the changes. Have you looked at image analysis as a way of providing mathematical analysis of the problem? Yeah. Absolutely,
and there's a lot of really quite sophisticated mathematical modelling and analysis that can be used on MRI. One of the problems is, though, is providing radiologists with a tool that they can use every day. So radiologists see like dozens and dozens and dozens of patients every day, may even be up to 100. So the tool that we develop has to be something that they can use within minutes and not sort of up to an hour per image, which is some of what the other sort of more complicated and sophisticated analysis techniques can involve. So we've tried to focus on a very simple technique that just looks at the area of bright signal, so that it's something that could be applied in a clinical setting just very quickly to many, many images every day, rather than trying to go for something that is really quite complicated and sophisticated, because that technology does exist, but it's just not in a form that is usable in clinical settings at the moment because of the time and the software involved. But it's a great suggestion. Thank you.
Thank you.
Yes, you spoke quite well about the degradation in movement and motor stuff. Is there a corresponding degradation in mental acuity, speed and accuracy, all that sort of stuff, mental nimbleness, or are the two separate?
No, they're not separate. So the disease process that affects the movement parts of the brain is also similar to the disease process that's affecting brain regions that are involved in cognition and memory and executive functionings and that sort of thing. The degree of symptom severity in that regard depends on disease stage and how much those other brain regions are affected. So some people with Parkinson's disease will progress to full-blown dementia as well because of their Parkinson's disease. But other people might only have mild cognitive impairment, something that's, not overly noticeable in their daily lives. It's still probably there if you teased it out in a research setting, but it's not enough to really impact their normal day-to-day living. So yeah, it just does depend on the disease stage. It's usually the later stages that are associated with changes in cognition and memory.
Any other? Florian.
Sorry, so you mentioned you're interested in identifying predisposition. As far as I understand, so I'm not a scientist. There's all this current work on genome-wide association studies, which essentially tries to use the full genome to kind of associate, okay, if you have these 500 little places, then you're more disposed towards developing it. Is that applied to Parkinson? And is there any kind of idea of how strong genetic influences versus environmental influences are?
Yeah, so there's probably 20 to 30 known genetic mutations that are evident in Parkinson's disease. Some of those genetic mutations relate to what's called familial Parkinson's disease. So that's Parkinson's disease that runs in families. But that's an uncommon form of Parkinson's. Only 15% of cases have a family history. The other 85 are of unknown origin. There's no family history. So what we think probably is happening is that there are some genes that can potentially mutate and that can lead to Parkinson's running in families. But in most other people, we think that there is some exposure to an environmental factor that changes the way that the cells are functioning, and some genes can mutate as a result of that. And so there are a cluster of genes that seem to be quite susceptible to potentially environmental insult. And it happens to be the genes that are really quite associated with a particular type of neuron in the brain, and that's the dopaminergic neurons that are quite vulnerable to that. So it's not only those genetic mutations, because the other changes in the cells can happen even without the genetic mutations. So it really is quite a complicated story, and it's why we still don't understand the full story yet, because there's a lot of factors involved. They all interrelate with one another, and it's difficult to pinpoint what's causing the changes to occur.
Thank you, David Lewis. I was going to ask about using AI, but I think you sort of answered that with the image analysis question. I was just wondering about the first stage where you mentioned that people lose their sense of smell, and I was wondering if there's any tests there that might indicate that could lead to Parkinson's, maybe with cell response or something like that.
Yeah, so there's been a lot of research done on the loss of the sense of smell in Parkinson's, and it's not isolated to just Parkinson's. So the dementias are often one of the first signs as well, is decreased sense of smell. And the thing is that it's really quite hard to focus on that as a diagnostic aid, because people don't realise they've lost their sense of smell often. You know, it's a gradual thing. And you don't know that a smell's there if you can't smell it. So often people don't realise that they have lost their sense of smell until they hit a neurologist's chair or a room where the neurologist goes, "Can you smell this?" And then they go, "No." Oh, okay. And then when you look back, it's like, yeah, they've actually been like that for a number of years. So there's really strong data that suggests that those neurons that are involved in the sense of smell, they're one of the earliest to go, and there's a lot of animal data that's replicated that as well. And it's those neurons that are particularly vulnerable to key environmental insults. In particular, environmental factors that have been linked to Parkinson's disease are the pesticide rotenone and the herbicide paraquat. So they're two chemicals that, if you expose laboratory animals, you get the same whole series of changes that occur in these neurons as you do in humans with Parkinson's. And the process, how the changes move through the nervous system, also has a lot of overlap. So sense of smell plays a really key role in the disease process, but it's hard to use as a diagnostic aid because it's not localised to Parkinson's, and people don't often realise that they have a problem until someone asks them about it.
Thank you. Got just two more questions, Robert Thomas.
Thank you very much, Robert Thomas. I was going to ask the same question, but because of observation, my older brother had Parkinson's and went on to Alzheimer's, and finally that was shortened his life and quality of life. But on reflection, you, as a family member, you could see a loss in mobility, but also in conversation from years before was the loss of smell.
Yep.
So people do know it. If it's sort of endemic with people taking drugs and so forth, is there a possibility that in your normal GP visit you could have a simple smell test or something? You know, like you need to cull people out and you need to bring people into the system.
Yeah.
And in the olfactory system, does that also come under some decay because of it's not being used properly? I mean, there must be a screening process that should be simple. The litmus test thing.
Yeah, there are tests they're actually called sniffing sticks, which is quite a sophisticated name. But essentially they're little white pen-shaped, nondescript. There's 12 of them, all of different scents. And yeah, you put each one under a person's nose and you ask them, what does it smell like, pretty much. So there are lots of easy-to-use olfactory tests that are out there. but they're not commonly used in GP clinics. That absolutely you wouldn't have your smell tested in a GP clinic. Some neurologists do them as part of exploration of potential for neurodegenerative diseases, but because the changes in smell aren't isolated to a particular disease, sometimes it just can help confirm a particular disease rather than point them in the right direction from the beginning. So if a person comes in where they're not moving properly, they've got a shuffling gait, they've mask-like expression, all sorts of things that you see in someone that has Parkinson's. You then do some imaging that would support it, as well as they've lost their sense of smell and have some gut disturbances. Then all of those together start to point to probably it's Parkinson's disease. So it's used within the diagnostic kind of scope, but it's not really strong enough to be an isolated test on its own.
Last question.
Okay. Your comment about paraquat causes me to ask, is it more prevalent, Parkinson's, among farmers?
Yes.
And the other thing is, presumably picking the disease up early is good for research, but does it do the patient a favour if it's incurable?
No. So the only thing that it does do is, I guess, opens the opportunity for monitoring. So for earlier monitoring and earlier management, and often that doesn't mean that they're going to get a better outcome, but earlier intervention is always better. The thing about earlier intervention as well, and earlier identification, is that if you can identify, well, actually I'm going to backtrack a little bit. So this brain region that we're focusing on, only once you get up to 80% of the cells dying in that brain region do you start to get movement symptoms. But if you can catch someone at 60% of loss of those cells, then they probably aren't even going to notice that they have any movement problems. And if you can keep them at that point for the rest of their life, then Parkinson's won't have a big impact on their life. But to find people that are at that point, you need early detection. Because there is a lot of research efforts being dedicated to drugs that are what we call neuroprotective. They're not trying to overcome the loss, they're not trying to make new neurons, they're just trying to keep the ones that are still there safe. And so if you could then, early detection, identify someone that's only lost 60% of their cells, put them on a new medication that helps to protect the remaining neurons, then they could live a happy and fulfilling life and never be really affected by their Parkinson's disease. But once you get to 80% loss of cells, there's not really coming back from that, because adult human brains can't make new neurons. So you can't replace the ones that have already died. So early intervention and identification does help, because it opens more opportunity to new drugs that are being developed.
Is there research on lizard's tails that we can use here?
Sorry?
Is there research on lizard's tails that we can use here?
On what, sorry?
Lizard's tails. Like regrowth?
Oh, lizard's tails. Oh, what are we talking about here? No, I'm not sure about that, but yeah. All right, thank you.
No worries. Thank you, Gabrielle. Just stay here for a moment. We want to thank you. Your talk has been very interesting, and I'm sure that each one of us has known somebody or of somebody who's had Parkinson's disease and how sinister it is. Gabrielle, we'd just really like to thank you for your talk today. We'd like to give you a certificate of appreciation and let you know that the money that we've raised today is going directly to Rotary Community and Youth Projects, and your presentation makes this possible. So thank you so much.
Thank you.